Once again, I'm here. My name is Deodat. Okay, I'm here presenting how to solve the questions of biology paper 1. And our paper is biology paper 1 2019. And in the beginning, I have already done up to question number 3. Today, I will continue with question number 4. And this paper, you're supposed to answer, it involves 2 sections, okay, section A and B. And student, they are supposed to, or the candidate, they are supposed to answer all questions in section A and only 2 questions in section B. So today, let me continue with question number 4. Okay, here. The question number 4, it says that, okay, the question says, define respiratory quotient. Define respiratory quotient, that is part A, and part B, it says outline the chemical changes which protein and fats undergo when they are used as energy source. Indicate where the products of these changes link up with carbohydrate respiration. Okay, outline the chemical changes which protein and fats undergo when they are used as energy source. Indicate where the product of these changes link up with carbohydrate respiration. Okay, let me start with part A, define respiratory quotient. Okay. For A, to define respiratory okay, the respiratory quotient here can be defined or can be referred as to the amount of carbon dioxide evolved divided by amount of oxygen. Okay, that is respiratory quotient. This is amount of carbon dioxide evolved, okay, divided by amount of oxygen used. Okay, so you have to define their candidate as, okay, this is just summary, then you can write like this one. So you have to define as this is the amount of carbon dioxide evolved divided by amount of oxygen used in the respiration. So the summary is as this one. Okay, this one, it is 1 mark over here. Okay, part B, it says the to outline the chemical changes which occur when protein and fats undergo when they are oxidized. Okay, oxidation of protein. Let us start with oxidation of protein. Okay. First one is that the protein, as normal, is enzymatically hydrolyzed to amino acid. Okay, is enzymatically hydrolyzed. Okay, this one is hydrolyzed to amino acid. Okay, this is normal digestion of protein, whereby the protein is hydrolyzed to amino acid. Then from there, amino acid, this one undergo deamination, it undergo deamination, that is removal of amine group in the amino acid to produce a product which is alpha keto acid. Alpha keto acid and ammonia. So the deamination, it undergo deamination whereby the amino acid, okay, is converted to alpha keto acid and ammonia. Then the alpha keto acid, this one alpha keto may be converted to pyruvic acid or acetyl coenzyme A. Okay, this one may be converted. Okay, may be converted to pyruvic acid or acetyl coenzyme A. Okay, so there are these three processes which is in the oxidation of protein. The first one is that it is hydrolyzed to amino acid. Then amino acid, it undergoes deamination to alpha keto acid and ammonia. Then alpha keto acid, this one is converted either to pyruvic acid or acetyl coenzyme A. And you know that as it goes to pyruvic acid and acetyl coenzyme A, this one is cooperated into a Krebs cycle. Okay, as normal in the respiration of glucose or of carbohydrate. So these are the three processes. So you student or you candidate, you have to explain it. So first of all, this is enzymatically hydrolyzation of amino acid. Then amino acid is changed or it undergo deamination to alpha keto acid and ammonia. Then alpha keto acid, this one is converted to pyruvic acid and acetyl coenzyme A. And this pyruvic acid and acetyl coenzyme A, as normal, it goes in the normal process of glucose respiration in the Krebs cycle. So that is about the protein. Okay. Okay. How about the fats? Okay, the fats. Okay, oxidation. The first one was protein, now fats. Okay, fat as normal is, this one is hydrolyzed. It is hydrolyzed to fatty acid and plus glycerol. Okay, and glycerol. Okay, this is normal digestion of fats. Okay, then the step number 2 is that the fatty acid undergo beta oxidation. Fatty acid, this one, it undergo, okay, the beta oxidation. Okay, the beta oxidation to acetyl coenzyme A. Okay, beta oxidation, this is breaking down of large or long chain of acid, fatty acid, long chain to 2 carbon chain, that is, which forms the acetyl. The acetyl has 2 carbon. So that is how The third one is that this the beta what the acetyl coenzyme A, this one enters. Okay, acetyl coenzyme A enters. Okay, the Krebs cycle. Okay, that is as glucose. Okay, as glucose. Then the glycerol here is converted to triose phosphate sugar, which is incorporated in the glycolysis. Okay, glycerol is converted. This one is converted. Okay, to triose. Okay, triose phosphate sugar. Okay, triose phosphate sugar, it means the sugar with 3 carbon. Okay, and that is this glycerol, which is there, is converted to triose phosphate sugar. And triose has 3 carbon, and 3 carbons, this is like a pyruvic acid. So, which is incorporated, okay, triose phosphate. Okay, this one is incorporated. It is incorporated in the glycolysis pathway. Okay, glycolysis pathway. So, because the question says, indicate where the product of these changes link up with carbohydrate respiration, whereby we know that glycolysis is a respiration of carbohydrate. Okay. So that is how this one is done. So the total, this one has total of 4.5, okay, marks. So each point, as we show it, it is 1.5 points. Okay, so you have to answer that way. Okay, let's continue with question number 5, dear viewers. Question number 5. The question says, what is meant by the limiting factor? Part B, okay, part A. What is meant by the limiting factor? From your knowledge of photosynthesis, this is part B. Explain precisely how the following factor can be a limiting factor in the photosynthesis. The first one, Roman 1, is temperature. Roman 2 is light. Okay, let us start with 5A. What is meant by limiting factor? Okay, this limiting factor, okay, is the factor which its presence is essential for the process. Presence of this one is, okay, is the factor, okay, whose presence, okay, is essential, okay, for the process. The second is that, okay, when it is below minimum level, the process will not occur. Okay, when occurs below minimum level, okay, process will not occur, even though even though all other factors are present. Okay, are present. Okay, in excess. So when you combine this, okay, this three, this is the factor whose presence is essential for the process. When occur below the minimum level, the process will not occur, even though all other factors are present in excess. So, in a short way, okay, or in a short way, we can say that this the rate of given process is determined by the factor which is in the shortest supply. Okay, short way. That the rate of given process, okay, is determined, okay, by the factor which is in short supply. Okay, which is in short supply. For example, assume that the carbon dioxide in the photosynthesis is there, it is enough, there is enough light, but the temperature is below the 0 centigrade. Okay, the temperature there will act like a limiting factor because all other, the process will not occur because of only temperature which is below. So that one is the limiting factor. So when you define this one, here you'll get 2 marks, okay? 2 marks, okay? You can define this, you can use this one or this one, okay? How now? How temperature, part B, 5B, says that how temperature and light acts as the limiting factor. Okay, temperature, let us start with temperature as limiting factor. Okay, in the photosynthesis. Okay, temperature as limiting factor in the photosynthesis. Okay, so this one, you can start by this one, by this way. The temperature, we know that photosynthesis is enzymatically controlled reaction. Okay, the photosynthesis is enzymatically controlled reaction, and we know that all reaction which is controlled by enzymes, it needs a certain optimum temperature. So when the temperature, is below or is below 0, is 0 centigrade Celsius. Okay, the enzymes will be inactivated, function well because of the temperature which is below the, will increase. Okay, increasing of temperature to optimum level. Okay, also the rate of photosynthesis will increase. Okay, this is okay. This is due. Okay, this is due to the increase of velocity of collision. Velocity of collision between the substrate and enzymes. Okay. So as the temperature increases, usually the colliding reacting substance will react with a very high velocity or colliding with very high velocity, okay, to produce the required products. Okay, so here it is said that in increasing of 10 centigrade Celsius temperature, okay, usually reactions rate also doubles. Okay, it doubles. So on increasing 10 centigrade of temperature below the optimum temperature, the reaction rate also doubles. Okay, that is how the reaction rate usually doubles. Then above optimum temperature, the enzymes okay, photosynthesis will be denatured. Okay, the enzymes will be denatured and the rate, consequently, the rate will decrease. The rate will decrease. Okay. Okay. That is how to answer how the temperature as a limiting factor. So we know that the photosynthesis is a controlled reaction. When the temperature is below 0, the enzymes will be inactive. Hence, the rate will slow down. As the temperature increases below the optimum temperature, the reaction, the photosynthesis will increase. And it is said that as you increase 10 centigrade, usually the reaction, any kind of reaction, doubles. And when the temperature is more above the optimum, usually the enzymes will be denatured. That is how. So let's go to another, how the light as a limiting factor. Okay, the light as a limiting factor. That is 5B Roman 2. light as limiting factor. Okay. So the first point is that under light is needed in the photosynthesis. Okay, in the photosynthesis to produce, okay, light. Okay, in the photosynthesis, okay, produces two things. Okay, produces ATP and nicotine amide adenine diphosphate dinucleotide phosphate, which is reduced. Okay, this is nuclear and nicotine amide is known as nicotine amide adenine dinucleotide. Dinucleotide phosphate. Okay, since it has hydrogen here, it is reduced. Okay, so it is reduced. So these two products are produced in the light depending reaction. Okay, this is in the light depending reaction. So on answering, you have to say in the light depending reaction, okay, light is important, but to produce two products, that is ATP and nicotine, the reduced nicotine amide adenine dinucleotide phosphate. That is the first one. Hence, okay, increase light intensity. Okay, increase light intensity will increase the rate of photosynthesis. It will increase the rate of photosynthesis because it will produce these two products in adequate number. So point number 3 is that also the light causes the opening of the stomatal pore. Light causes opening of stomata pores. Okay, if the stomata is open, there is that diffusion of carbon dioxide, which is required in the dark reaction in the photosynthesis. Okay, which leads to increase of diffusion in of carbon dioxide. Hence, increases the rate of dark reaction, which fixes, okay, carbon dioxide to carbohydrate. Okay, that is about that one. So, as the light increases far above, okay, too much light causes the stomata pore to close, hence it decreases the rate of diffusion. Too much light okay, decreases. Okay, too much light okay, causes the closing of stomatopore. Stomatopore. Okay, it causes the closing of stomatopore. When it is closed, the diffusion of carbon dioxide will decrease. Hence the rate also decreases. So closing of stomatopore, which reduces carbon dioxide diffusion. Okay, okay. This one, hence the rate the rate of photosynthesis will also decrease. Okay, so when you mention those 4 points, each point has 1.5 marks. Okay, so each point has 1.5 marks, and you're supposed to explain like those 4 parts. Okay, so you'll have 4.5 marks. So in the beginning, we said that the light is very important because it produces ATP and reduced nicotinamide adenine nucleotide. Then increasing the light intensity will also increase the rate of reaction because of production of that ATP. The light causes opening of stomatopores. Okay, this leads to the diffusion of carbon dioxide and hence fixation of carbon dioxide to carbohydrate. Lastly, the fourth one is that the carbon dioxide, as light intensity is too much, the closing of stomata will occur, and that one will decrease the diffusion of carbon dioxide, and hence it also decreases the rate of photosynthesis. So that is question number 5. Let's continue with question number 6. Question number 6. The question says, asexual reproduction produces offspring which is identical to their parents, where sexual reproduction produces variation. Discuss this statement. That is the first part, part A. Part B, draw a well diagram of human spermatozoon. Okay. Let's start with part A, which says sexual reproduction, identical offspring, but as a sexual reproduction, it produces variation. So asexual reproduction, okay, with identical offspring. While sexual reproduction has variation in offspring. Okay, that one. Then the second is to draw the diagram of human spermatozoa. Okay, let's start with this one. Okay, this asexual reproduction is reproduction. This asexual reproduction is reproduction. This is reproduction which involve a single organism which do not produce. Is a reproduction involves single organism, okay, without production of gametes. This does not produce gamete. It involves a single organism. An example of this one is like binary fission, e.g. binary fission, okay, vegetative propagation, okay, like that one. An example of that one. This asexual reproduction, this one involves mitotic division to produce a daughter cell. Okay, this one involves mitotic division, okay, to produce the daughter cell. Okay, the daughter cell which is similar, daughter cells, two daughter cells which are similar to the parent cell, okay, which are similar to the parent cell. So there is no any involvement of other organism. It is self, they divide, so it will produce this similar offspring. Okay, that one. And on the other hand, okay, on other hand, sexual reproduction, the sexual reproduction, this one involves meiosis to produce haploid. This sexual reproduction involves different parents, okay, of the same species. Of the species, okay, by production of haploid gametes. during meiosis process. Okay, during meiosis process. So the sexual reproduction, it involves two parents, okay, to produce the haploid gametes during the meiosis. And why it has variation? Okay, why this one has variation? Why variation in the sexual reproduction? Okay, when you mention like this one, a sexual reproduction, you define it like that one, you'll have one mark here. Okay, what is asexual reproduction? As you mentioned, asexual reproduction, it involves meiotic, you'll have one mark. And now as you continue with the sexual reproduction and telling why it has the variation, okay, because it involves meiosis. And okay, the variation in sexual reproduction okay, is due to the first one. There is independent assortment. Okay, independent assortment of chromosomes. Okay, this one is, there is a random movement of chromosomes during meiosis 1, whereby the homologous, chromatids of homologous chromosomes arrange and separate randomly. Okay, where here, okay, there is a movement. There is random movement of chromosomes. Okay, in the meiosis 1. Okay, random movement of chromosomes in the meiosis 1. That is why it causes that variation. So let me show like this one. Okay, when you're answering this question, there is no need, the question does not need you to draw the diagram which I'm going to show. But if they say illustrate it with the diagram, you have to use the diagram which I'm showing you. Okay, the independent assortment. Okay, assume your the cell is like this one. Okay, there is these chromosomes here. Okay, with another one here, and another one. Okay, during meiosis 1, these two are homologous chromosomes. This one. The separation during meiosis 1 can be like this one. There is formation of a cell which looks like this one. Either there is all these these chromosomes may form this one and this one. That is a single gamete. And another one will form, okay, like this. Okay, if it is that way, this another one will be like, so there is this formation of gamete like that one. Or maybe like this one. Okay, this homologous, okay, the black one, this one, and this homologous chromosomes, and this one. So, as you can see here, there is either the independent or segregation may occur like this one, or may occur like this. This is black and black in the single cell, the gamete, and the red and red in the single gamete. Or the segregation or independent assortment may occur like this one. There is black, okay, this black here, and another red, and the black and the red, like that one. So this causes variation in the offspring when they produce offspring. Okay, another cause of, so you're not required, according to this question, you're not required this one. Okay, this is just to show you, in order for you to understand. Another cause of variation is due to the crossing over. Okay, another cause is due to crossing over. The crossing over. This crossing over occurs in the during the homologous chromosomes. Okay, during meiosis 1. Okay, in the diploid stage. Okay, here in the crossing over there is that exchange. There is formation, chiasmata formation, and there is a sweeping. Okay, it says like this. There is it occurs by chiasmata formation between chromatids of homologous chromosomes, and the breakage occur causing the gene swap from chromosomes, leading to new gene combination in the resulting chromatids. So the crossing over of homologous chromosomes result to the chromosomes with a new gene combination by crossing over or by chiasmata formation, and then breakage, and the new chromosomes which will be formed has a new combination of a new combination of that one. The third one is due to the random fusion of gamete. Random fusion of gametes. Okay. For example, in the single gamete, single female gamete can have a single female gamete here can fuse with any male gamete during fertilization. Okay, e.g., single female gametes may fuse with any male gametes, okay, in their fertilization. So the fertilization may occur randomly. We know that usually there is millions of male gamete which compete in the single female gamete. For example, in the case of human male. So any gametes, male gamete, may fertilize that one. So this one is called the random fertilization. This leads also to the variation. Okay. Okay. Part 2, it says to draw the diagram of human spermatozoon. Okay, 6. The diagram of human spermatozoa. Okay, the diagram of human spermatozoa. Okay, the diagram will look like this one. Okay, it will look like, okay, like this one. Mm-hmm. Okay, this one is acrosome. 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 And this one is nuclear. This is mitochondria. This outer one is cell membrane. Cell membrane. And there is this centriole. Centriole, since it's one centriole. Okay. The centriole there. Okay, here, this one to this point, this is head, and here to this point here is the neck, This is middle piece. Middle piece. From here, going to this part is what is known as a tail. Okay, so when you draw a good diagram which looks like that one, the caption, you have, the caption is one mark, then diagram, a good diagram that will carry 2 marks, and the labeling. Labeling, you're supposed to label 4, so it will be 2 of that. That is how you can do in this one. Okay, let me finish here today.